Our next speaker is Dr. Shuvo Roy, who comes to us from the University of California at San Francisco, where he serves as a professor of the Department of Bioengineering and Therapeutic Sciences and Surgery. More importantly, he serves as the head of the Kidney Project, where he works alongside Dr. William Fazell of Vanderbilt University on an artificial implantable kidney. He's quite the patient advocate, a very good friend of AAKP, and we actually partnered as an organization with the Kidney Project in 2018 because the American Association of Kidney Patients believes so strongly in the advancements of science and innovation and for a future that includes artificial implantable organs, namely the kidney. Dr. Roy, go right ahead.
Thank you, AAKP and George Washington University, for the opportunity to provide an update on the Kidney Project. This update will cover some of the recent developments as well as the background on the Kidney Project that has set as its goal the development of a new technology to treat kidney failure. As you can see, our goal is to develop an implantable bioartificial kidney. What is this? This is a compact device that is surgically implanted in the abdomen to provide continuous treatment, and it combines a hemofilter unit to remove toxins from blood with a cell therapy unit, which we call the bioreactor, to provide the other primary functions of a healthy kidney. It operates continuously, as I have mentioned. Because it's implanted, there are no external connections. It allows for total freedom of mobility. By avoiding the through-skin connections, we minimize the risks of infection from outside. Because the bioreactor has kidney cells, it will provide more physiological therapy than dialysis would. And because the cells are protected inside the device, the patient's immune system and the cells are separated from each other, which means there's no need for immune suppression drugs. And the device can be instrumented with sensors to not only monitor its function, but also report on the status of the patient's health. The idea of the implantable bioartificial kidney is that it would get surgically implanted through a surgical procedure not very different from a kidney transplant, connected to the blood vessels, and then the waste would be directed to the bladder to pass out as urine. And this vision of the bioartificial kidney has driven us over the last decade to advance the technology to make this happen. The work we are doing here is building on the pioneering cell therapy work that was conducted by Dr. Humes at the University of Michigan in the late 90s and early 2000s. What he did was to actually take conventional dialyzer technology, dialyzer filters, dialysis machines, except he connected two of them together to mimic the architecture of the kidney's nephron. The filter to remove the toxins in the kidney is called the glomerulus. Here we call it the hemofilter. Blood comes into the hemofilter, generates ultrafiltrate, which is then directed to the next stage of the device, which we call the bioreactor, which mimics the kidney's tubule. The bioreactor is lined with kidney cells that perform the primary functions that a native kidney does, and these two components together provide more complete renal replacement therapy. So we are mimicking the architecture of the device that Dr. Humes developed over 2 decades ago, but making it available for the larger community that needs to be treated continuously and at home for kidney failure. So this is the fundamental question that we've been working on, is how do you take the renal cell therapy work that Dr. Humes did, which is being implemented here on the left side of the screen, and make it an implanted continuous therapy device for the vast majority of chronic kidney failure patients, which is shown on the right. And this is what we call the Kidney Project, and that is the effort that has driven our team over the last decade and a half. And the approach we are taking is an engineering solution. What I mean by that is that we are taking the known knowledge that's out there and meticulously integrating it, refining it, optimizing it to create a device that can provide therapy. We took this approach because, unlike discovery science, which requires elements of chance and requires a lot of fundamental discovery time to get to a meaningful result, the engineering solution allows us to bring a talented team together and get to a solution that can provide treatment in a shorter period of time than most discovery science-based projects. Here you see a slide of a rover that will go to Mars and a team of engineers and technicians putting it together. It does take time. It does take resources in terms of money, but fundamentally, once it's done, it's going to go to Mars and it's going to work. It's not waiting for a new principle of physics or chemistry to be discovered. And we contrast this to some of the other basic science approaches that may result in a breakthrough, no doubt, but may take a much longer time to get to patients. The fundamental technology we are using to miniaturize the concept of the renal cell therapy device that Dr. Humes demonstrated at the University of Michigan over 2 decades ago is what we call silicon nanopore membranes. This is a technology that has been adapted from semiconductor microelectronics. The same technology used to make your phones, your laptops, and the like, and you can use this technology to precisely create a new membrane geometry that outperforms the conventional membranes that are used in dialysis today. Because of the precise geometry, you can have a membrane that is highly selective. Toxins from blood, just as your normal kidneys would do it, and it's highly permeable. What that means is that it requires very little energy to achieve that filtration, such that you actually do not need electromechanical pumps. You can run this just off the body's own blood pressure alone. And because it's silicon that can be modified on its surface by surface chemistry techniques, you can make it highly biologically compatible. So this technology, the SNM, as we call it, allows for a biomimetic filter unit that can operate with just the body's own blood pressure without the need for significant energy driving requirements, and can be operated for a long time inside the body because it's biocompatible. Now, I will take the opportunity to share a video. This video will showcase some of the research highlights that my team have worked on over the past few years. This video is from the lab of my colleague, Dr. William Fizel at Vanderbilt University Medical Center, and it showcases the research work that has to go in towards the development of an artificial kidney, in this case, both the mechanical filter and the cell therapy unit. Please watch.
The future is looking brighter for patients with kidney disease. Not only are new medicines and treatments slowing progression of disease, but new treatments for kidney failure are coming soon as well. Researchers have made breakthrough progress growing kidney cells in the lab for cell therapy of end-stage kidney disease. Long before researchers grow new kidneys in the laboratory or transplant them from animals into people, a combination of cutting-edge nanotechnology and living kidney cells will free patients from the tether of dialysis and provide the benefits of kidney transplant to everyone with kidney failure. Let's see how this universal donor kidney works. Kidneys filter the blood to separate wastes from healthy blood and then concentrate wastes from the whole body into 1 to 2 liters of urine each day. The biohybrid kidney does exactly the same thing, unlike dialysis with its heavy machines and batteries and gallons of water, which can never be implanted.
The cells that make up the kidney's filter are extraordinarily fragile, and leakiness of the kidney's filters underlies almost all of the kidney disease that results in kidney failure and the need for dialysis. Those cells are exceptionally fragile. They do not grow in the dish.
So Dr. Fizel and his team designed a new kind of artificial filtration membrane based on the size and shape of healthy kidney cells. This membrane is the first completely new technology in renal replacement in over 25 years. The collaboration with Dr. Shuvo allowed this idea to progress from concept to FDA application. However, it's not just the kidney's filter cells that misbehave. Most human cells really don't look or act in a lab dish the same way they do in a healthy person. The tubule cells that concentrate wastes from the whole body into a little bit of urine don't reabsorb salt and water and don't excrete toxins when grown in an artificial environment.
This isn't just a problem for artificial kidneys. Drug companies can't use cells in a dish to test for efficacy or toxicity.
And medical doctors have to use entire animals to examine mechanisms of disease and effect of treatments. Solving the problem of cell culture stress solves many problems. Dr. Fizel felt that part of the problem was that cell culture was vastly different to a healthy body. Plastic lab dishes are hard, human tissues are soft, kidney cells have fluid flowing over them. Cells in a dish are still. We culture kidney cells with serum, but in the body, the cells don't really get exposed to serum. Dr. Fizel started testing these simple factors one by one and in combination.
Testing these factors, it's like a centipede driving a bus. If there's 75 feet on the brake pedal and you take 1 foot off, the bus still doesn't go forward.
The cells certainly look different on soft materials than on hard plastic, but they didn't seem to reflect these changes in the proteins they expressed, the key proteins. Enter serendipity. Rachel Evans runs the cell culture in Dr. Fizel's lab, and she had tested one batch of cells seeded on soft materials to see if they were making proteins to reabsorb sodium, and they were not. However, she had kept a second batch of cells to test if something went wrong with the first tests. She kept changing the cell culture medium on them until a few more weeks had gone by. Far longer than most people grow this kind of cell in the lab. She ran the test again on these cells that had been in the dish for weeks, and the cells had started to make the sodium exchanger proteins, a basic function of natural kidney cells. From there, the team went on to examine the signaling pathways that governed the response to soft versus stiff materials. By manipulating those pathways with drugs, they were able to coax the cells into pumping salt and water just like they do in a kidney. From the start of the project, we have tried to improve on what we accomplished to strive every day for a better life for patients with kidney failure.
70 years ago, we treated polio with an iron lung, but medical advances have put the iron lung out of the clinic and in a museum, and we strive to do the exact same thing for dialysis machines.
I hope you saw the range of disciplines, the amount of work it takes to build components of the bioartificial kidney. In this slide, I focus on just the mechanical part. You may recall the little silicon membrane that was referenced in the video. Here, we are now talking about moving it from a small filter unit towards something that would get closer to patients. On the top left, you'll see a photograph of a mini prototype of a filter. And on the right-hand side is a schematic showing where blood goes in and out and where the ultrafiltrate comes out. And we have spent time to design, optimize this device so that it can be operated in the benchtop and preclinical settings. Here, we've actually implanted that device into this animal, and you see a catheter sticking out. So we can test the performance of the filter after implantation. We ran that filter through a dialysis machine on the outside. Note, the filter is already implanted. The little tubing that you see is the catheter that goes to the dialysis machine. And we are able to run dialysis in this animal over the course of a few days. Some key points. There was no external blood pump. There was no blood outside the body, but we're able to perform hemodialysis. We did not need to use systemic anticoagulation like heparin. The design of our filter and the coatings of the filter are optimized such that the blood flows without causing thrombi or blood clots. And as such, when it was not connected to the machine, the pig, in this case, was able to move freely and drink freely. And what you see on the right is the data over the course of 3 days. And you should note there's no loss of albumin, no loss of protein over those 3 days. We did have a urea and creatinine clearance that is comparable to commercial dialyzer filters. But again, we did not use a blood pump. And there's a slight decrease on the 3rd day for clearance. This is very exciting for us because it shows us that we can make a filter that can operate just under the body's own blood pressure without systemic anticoagulation and provide toxin removal from blood, making us excited that we can build the hemofilter component of the bioartificial kidney. So next we looked at the cell therapy unit, which we call the bioreactor. You see a schematic of the bioreactor on the left and being implanted on the right. And what you're seeing in the middle are the cells that are put into the bioreactor. Each of the little gray squares that you see on the right-hand side are human kidney cells that are isolated from a cadaver kidney and grown in culture and then inserted into this bioreactor device. We put the bioreactor device into the animal and closed up the animal. And the cells in the bioreactor were kept in the animal for 3 days. In this case, we implanted the bioreactor in the region in the neck so we could monitor any potential complications readily via ultrasound. And what you see on the right is the cells have grown and are alive after 3 days of implantation. Why is that exciting? We've actually put human kidney cells in our device and implanted that into a pig. And we did not need any immunosuppression drugs. If we did that without our device, all the cells that we would implant would die because the human cells and the pig's immune system would reject them. But our membrane has the capacity to provide immunoprotection. And by protecting the cells, you can avoid the need for the anti-rejection drugs. And just like the hemofilter, we again did not need heparin or other anticoagulant drugs, and the device operated just under the pig's own blood pressure. So we now have taken the advances that we've learned from the filter by itself and the advances we've learned from the bioreactor by itself. And now we have embarked on the next stage in our journey. That is to integrate the filter unit with the bioreactor unit. What you see here is one of our first prototypes showing the filter and the bioreactor integrated together into a prototype implantable bioartificial kidney. The top half is the filter, the bottom half is the bioreactor. And there's silicon membranes, the S&M, and the cells to provide both filtration and cell therapy. I should stress, this is a prototype and it's a mini-scale prototype. It does not have enough membrane area or enough cells to provide sufficient treatment for a kidney patient yet. So there's more work to be done, but it shows that we can actually build them and integrate them together. Here we show the timeline of the kidney project from early stage conceptualization in the early 2000s, where we basically looked at the work that was done with the external renal assist device, the renal cell therapy device. We developed the silicon membrane technology and showed its proof of concept. And then we moved into preclinical work and optimization, where we deconstructed the project into a bioreactor development portion and a hemofilter development portion. And you can see for the bioreactor, we took on 3 key tasks. And for the hemofilter, we took on 5 key tasks that were successfully completed last decade. And now we are finishing up the bioreactor and moving into the integration phase. It is not trivial to integrate, but we'll have to integrate the 2 components and also scale up the device. What does that mean? It means we have to put sufficient cells and sufficient membranes to provide renal replacement therapy. It's not discovery science, it's engineering optimization. It does take time, and there will inevitably be areas we need to refine our work, but we are very hopeful that these are all tractable issues. Once we've shown that we can integrate the device in us and provide therapy to a preclinical model that has compromised kidney function, then we can move on to thinking about the first human tests. I show this slide and also point out the challenges. One has been the unusual past year of the pandemic. It has impacted our supply chain, it has impacted the pace of progress, and we are very fortunate that now we're beginning to come out of that and ramping up the pace of our work. We're also challenged by the need for funds. This work is obviously meaningful to many of the patient community, many of the physician community. Unfortunately, we are not at the stage that we are readily accessible to the investor community because they feel like we need to develop our technology further and demonstrate its feasibility in humans. But we are also challenged by being further out than the basic science proof of concept, which is where traditionally research projects get their grant funding either from NIH or National Science Foundation. So we try to work creatively with funding agencies, with donors, with patient groups like AAKP to encourage the resources to come to projects like ours so we can get this done and completed and advance towards patients in the coming years. I will end by saying that the progress we've made to date is due to a number of resources and people. So we have received early stage funding from the National Institutes of Health and other federal agencies. We've been fortunate to also been able to attract foundation funding and also donors, small dollar donors to some larger dollar donors that have continued to support us to move the project forward. But we need to be able to get this done. To get this done, my colleague, Dr. William Fizel, shown on the top right, and I encourage all members of the AAKP community to reach out to the various stakeholders and encourage them to support mechanisms that allow us to complete the development of the implantable bioartificial kidney. We have a great team. I encourage you to check out our website, and thank you so much for this opportunity.
Thank you very much, Dr. Roy. AAKP enjoys our partnership with the Kidney Project and your longtime support for our efforts here at the Global Summit and as the nation's largest kidney patient organization.